Welcome everyone joining in. This is the North Central IPM Center Pests and Progress webinar series. Welcome to today's webinar. Our webinar is titled From Detection to Action, eDNA, 3D Printing, and Grower Outreach for Spotted Lanternfly. I just want to introduce myself. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center, where we are one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. We have a mission to coordinate IPM across our region by sharing and promoting IPM research, extension, and teaching activities. If during today's webinar you have any questions, there's a Q&A portion at the bottom of your screen. Please type your question in there and we will answer them after the webinar is completed. So I'm excited to introduce Ashley Leach and Natalie Constancio, both from The Ohio State University, who are joining us from a classroom and will talk to us all about spotted lanternfly. So Ashley, I'll let you take it away.
Yeah, thank you, Laura, for that introduction. Hopefully you all can hear me. And I appreciate those who are tuning in during, I'm assuming, a very busy field season. I know that we are certainly kind of going gangbusters over here, so I'm sure that's the case with many of you. So thank you for that. I am the state specialty crop entomologist here in Ohio. And so my interaction with this pest has been very diverse and nuanced because we are certainly working on this pest in grape production, but we've also been working on it a lot in urban settings as well. So we'll touch on some of that research today. And I want to talk about these 3 topics here. The first is I want to give you just a primer and introduction on spotted lanternfly, talk a little bit about its impacts in grape production, and then I am joined by the wonderful Natalie Constancio, who will be switching spots, and she'll be talking about spotted lanternfly in Ohio. So many of you may have been confronted with images similar to this. So this is a video taken by Christopher Jordan, and he's showing a infestation of spotted lanternfly on a backyard tree. And this video and many of these images have created this media fervor surrounding spotted lanternfly. And we are often, at least in my position as extension, as primary extension appointment, I'm often dealing with stakeholders and residents that might be contending with the impacts of this pest. So I think the first thing we need to do is kind of slow down and decouple, what we're dealing with in our backyard and what we're dealing with in our crops versus what we might have heard on Facebook. So with that, I want to kind of provide a brief primer here. So spotted lanternfly is a plant hopper insect. It's in the family Fulgoridae for those bug nerds in the room. This has been in the United States for well over a decade now. It was found in Pennsylvania in 2014. However, the Pennsylvania Department of Agriculture estimates that it's actually been in the United States for probably since 2012. And one thing I like to bring up is that it has become invasive in other places in the world, including Korea and Japan. And so I think this is an important place to kind of sit and remember that there's a lot of other places where we can gather literature in terms of impacts and also kind of understanding what other countries and places have seen when it comes to spotted lanternfly impacts. And it's been very successful in these different habitats where it's become invasive due to a diversity of reasons, one of which is that it can take advantage of over 100 different plant hosts. So it has a very varied diet, including what you're seeing in the background here, which is Tree of Heaven. So that is an invasive plant here in the United States, and it's one of spotted lanternfly's favorite foods, in addition to other things that we'll be talking about, like grapes and backyard ornamentals and hardwood species. Now, the other reason why spotted lanternfly is pretty successful is because we also have different types of appearances of spotted lanternfly. And as you can see here, the egg mass, which looks pretty indiscriminate from other things in the landscape, it is often kind of missed. And Natalie will be talking about that in a little bit, what that really might imply for its success. in moving across the country and becoming an issue in other places. But that's the egg mass stage. This is where we're going to see spotted lanternfly over winter. And then, at least in Ohio, we typically see emergence of these egg masses around May into June. And then we have our first through third instars, which have this black body with white polka dots. And then we have our fourth instars that show up late June into July, and those have that red and black body with the white polka dots. And then finally we get our adults, which show up in August, and they stick around for a while. And I'll actually talk about what that phenology looks like in some of our vulnerable specialty crops, in particular grapes. So the reason why, of course, I care about this and why I'm talking about this is because spotted lanternfly was found in Ohio in 2020 in Jefferson County, which is right up against the border of between Ohio and Pennsylvania. And I have my position here as a state specialty crop entomologist beginning in January of 2022. So when I started, we had 2 counties with active infestations of spotted lanternfly, which is what you're seeing on the map here. Now, this is a map that's put together by the New York IPM New York IPM group. And so if you're interested in this type of information, I definitely recommend you go ahead and visit that website, and I'd be happy to drop the link in the chat at the end. It shows you kind of the spread of this pest. And so when I started, we had these 2 counties, and then I fast forward a year and we doubled that. We have 4 counties, and then fast forward another year, we actually quadrupled that. And now we're at a place where we've kind of stopped counting because the entire state is under quarantine. In February of 2026, we actually, the ODA, the Ohio Department of Agriculture, issued a statewide quarantine for spotted lanternfly. And what we're seeing is that this progression of spotted lanternfly moving westward follows a lot of the models that have suggested, how exactly SLF is going to move in the coming years. I'm going to feature this POPS model that was developed out of a group from NC State. However, there's lots of other wonderful modelers that have produced similar models that I definitely encourage you to take a peek at if you're interested. But if we go back in time, we see, of course, that the spotted lanternfly infestation fairly contained to Pennsylvania. Fast forward a couple of years to where we are right now. In kind of 2026, 2027, and we see that there's a lot more movement of spotted lanternfly. And the kind of showcase in this paper in particular was that we're going to expect to see spotted lanternfly in California predicted by 2033. So the primary covariates that get highlighted in this case and many of the other modeling efforts are 2 things that are facilitating the spread of spotted lanternfly. The first of which is transportation corridors, and this really speaks to the fact that spotted lanternfly is human-mediated transportation or human-mediated movement. So that's how spotted lanternfly is really getting from place to place. And so when people talk to me and they ask me, okay, how do we get SLF to stop? How do we eradicate spotted lanternfly? I kind of jokingly say, well, we need to get people to stop moving, because that's how we're really seeing SLF move, is through these transportation corridors. We also know that spotted lanternfly, when it hops off that transportation corridor, whether it's a car or a railway or a rail car, we know that there is a wide variety of plant hosts that SLF can take advantage of, including grapes. So grape production has been something that many are concerned about in the face of spotted lanternfly movement, and that's because we're consistently seeing negative impacts of spotted lanternfly feeding on grape production metrics, and I'll talk a little bit about what that is. So in this case, this is just a correlation that we published over 5 years ago now, showing you that as we increase the number of spotted lanternfly adults feeding per vine, we're seeing a subsequent decrease in the clusters per shoot. Now, there's been a lot more research on this impact in the past couple of years, and there's more to come. And from these different publications that you can see here, what we're seeing Overall, so these are kind of general trends, is that more spotted lanternfly is correlated with reduced photosynthesis and sap flow, sugar content of fruit, cold hardiness of potentially the overwintering success of these plants, and then of course the yield in the following year. So knowing that spotted lanternfly can take advantage of a crop like grapes, it can also take advantage of wild grapes and many of the backyard trees we might have, in addition to tree of heaven. And this is particularly important in the Midwest, where we have a large amount or a large acreage of grapes throughout the Midwest, including Ohio and Michigan and other states. In many ways, Ohio represents this corridor as we're moving from the Northeast, where we have a large amount of spotted lanternfly infestation, into the Midwest. So thinking about management is really where we are sitting right now. So how do we figure out how to detect this pest and then how to manage this pest? So back in 2019, I actually was able to participate and contribute to spotted lanternfly research in Pennsylvania. And this is because at the time, my sister Heather Leach was the Specialty Crop Extension associate at Penn State. And so she was looking at a lot of these different management efforts in vineyards. And so we kind of thought, well, we kind of need to back up the bus a little bit, because we need to think about how we're building an effective pest management program. And we thought, well, we need to answer these two questions. When is SLF and where is SLF? And I kind of already hinted at some of this already. So I'll talk more about what we did specifically in these vineyards in Pennsylvania. So this is right in the heart of the infestation that we initially saw that outbreak in Pennsylvania. And we conducted these weekly scouting reports, or we had these transects that were walked weekly throughout the growing season across 2 years. And so kind of sharing the phenology here, each of these colors corresponds to a different instar. So there's 4 instars of spotted lanternfly. And what you see is that initially we have large populations of first instars, about 5 to 6 first instars per vine. And then as that SLF ages, we're getting fewer and fewer. But the biggest thing that we have to contend with in the vineyard is the adult population, which you can see here in orange. We see that this adult population is about 3 times greater than any of the nymphs that we saw. Like the greatest nymph density would be about 5 to 6, and we're seeing an average between 14 to 16 adults per vine in September. So this is really where when we think about management of spotted lanternfly, we're thinking about right now. What are we going to do about spotted lanternfly in the fall? So in addition to that, we wanted to understand how SLF was spatially organizing itself in the vineyard system. And that's where we did some grid sampling in addition to some spatial statistics. And that's really, my sister took the heat of that, of doing all of the spatial statistics related to this. We went out and we grid sampled spotted lanternfly at 2 different times throughout the season when they were basically in the adult, so that September, October window. And the idea was to be able to have a sense of what type of spatial distribution we were seeing for this pest. So we know that our monitoring efforts and also our management efforts are somewhat related to how that animal, how that pest is organizing itself. So we can have a organization pattern that looks something like an edge effect, where we would expect the most amount of pests along the edges of a field or a block. We might have a uniform distribution or a clump distribution that's fairly random, and we get these kind of hot spots throughout the field. And so jumping right into the results for this, what we see is that there is some variation in these different vineyard blocks. So this is just a subset of some of these vineyards that we looked at. And so all you're seeing is the hotter the color, the more SLF were there. And so each kind of box here on the edges, you see more of that red color, suggesting more of this edge effect that we're kind of seeing shown here on the left-hand side. So this is a really common spatial pattern for different types of agricultural pests. And coupled with the fact that spotted lanternfly adults are really good at dispersing and moving. We see that they're moving from the woodlot to the vineyard block often, and they're kind of organizing themselves along the edges of these two kind of landscapes, or these two different types, these two different features in the landscape. We see that this pattern also is shown in the scouting data, where the majority of spotted lanternflies were found within the first 18 meters of these vineyard blocks. So it gives us a good sense of really the management is going to kind of hang around these peripheries. And so looking at these two different patterns here, we have when is SLF, and this is kind of what I already mentioned in the very beginning of the presentation. We know that spotted lanternflies adults are becoming particularly problematic right about now. And this is true not only in urban areas, but also in our vineyard systems where we conducted this data. Additionally, we know that SLF is primarily found around these vineyard edges. But the next piece of this is looking at what we can do with this information now that we're in Ohio, where we don't have this type of outbreak. We don't have vineyards that are incredibly infested with spotted lanternfly. We're actually able to kind of rewind the clock and ask questions that we might not have been able to ask originally. And those are things like how exactly we're going to be able to detect spotted lanternfly. And so being in Ohio, we see spotted lanternfly somewhat as a looming threat on the state, because we have a number of different favorite host plants, as you can see here, but most concerning, of course, is the grape acreage that we have within the state. Ohio is a number 6 producer in the country for wine. We have between 1,000 to 2,000 acres of grape, as you can see here. And this acreage supports over 400 wineries. In a recent economic analysis, this industry was valued at over $2 billion. And so suffice to say, there's a lot of vested interest in maintaining the health of these vines and making sure that we can protect them against pests like spotted lanternfly. And I'm going to argue that this is not a unique story in the Midwest. We have many states that have a robust grape industry that we should protect. So now we get to be in this position of figuring out how exactly do we detect SLF and prepare for this incoming infestation. And so I'm going to go ahead and pass the mic over to Natalie, and she'll talk a little bit about our specific work that we're doing in Ohio.
Thank you, Ashley. Hello, everyone. We're happy to have you here. I'm just going to go ahead and jump right in on how we are learning to detect SLF, especially here in Ohio when we are in those early stages of our invasion. And we have a lot of different monitoring and detection methods, and they range pretty wildly from passive to a really intensive monitoring strategy. Our first ones are going to be kind of these passive methods. If you haven't heard of circle trapping or protected sticky bands, what they are, what makes them so attractive to a lot of our growers is that you stick them on the tree and we take advantage of this upward movement of SLF. We know that they start at the bottom of the tree and then they walk upward. In this circle trap, they go through the funnel and they're caught in the bag. In this protected sticky band, they walk straight up the tree and then they're stuck on the band. And we get to kind of keep track of whether or not there is SLF in this area. One of the downsides of this type of trapping is that you're really heavily reliant on choosing the right host. So we know that they love Tree of Heaven. And so a lot of times we stick the traps right on this tree. But again, your trapping is only going to be as good as your ability to pick that host. And so here's an example of that SLF movement that I was just talking about. Wrapping those traps around the correct tree trunk is going to give you a really good idea about whether or not you have SLF. Our next kind of method that Ashley just touched on was that active in-person scouting. So going out to your vineyards and your vines and your wooded area and taking the time to examine your vines and your trees and all of your plants for SLF. We do know that this takes a lot of time, it takes a lot of effort. It is helpful knowing that SLF is an edge pest because you do know where to concentrate your efforts. But when we have so much grape acreages and a lot of these vineyards that we visit, they're big, they're not backyard vineyards. We're looking at 100 acres. That's a lot to be walking for any single person, especially if you want to go out multiple times a week. And then our most labor-intensive sampling and monitoring method is eDNA collection. We already know a bit about spotted lanternfly, but we're going to talk about eDNA and how that works. So eDNA is predicated on this idea that as SLF is feeding, it is releasing honeydew, which is that sugary water, basically it's poop. And what that does is it leaves behind trace parts of the DNA. And so what we can do is then go through the vineyards, go through those vines, those trees, and sample, search for some of this eDNA using what we would call like a paint roller method, which is really us. And by us, I mean it's me walking the vineyard and I'm rubbing kind of just a paint roller like you would when you're painting your own home along that tree line, along those vines, picking up any of this sticky, sugary water. This is not something that we invented. This has been done by the Lockwood Lab. We know that this method works really well for detecting invasive pests. And in fact, we have published a study on this, specifically looking at that eDNA versus in-person scouting. And this was done in Cleveland, so a really metro area, really urban. We have our eDNA in the red and our scouting in the blue. And as you can see, just in general, our eDNA really outperformed any sort of traditional in-person scouting. And really, it beat it by about 2 times the magnitude. So about 60% of our samples were positive when looking at the eDNA versus only about 20% accuracy when you're doing that traditional in-person scouting. So again, why do we as specialty crop entomologists care about not only SLF, but these different trapping methods? And how do we prepare our growers to deal with this threat? And we do it by developing a scouting network across the state and teaching our growers to search for what isn't there. Because again, SLF is very cryptid. It's hard to find those egg masses. You can't exactly see eDNA either. So a lot of this methodology of the scouting can be really difficult. Ashley already showed you this map of our grape acreages across Ohio, but what I want to highlight is our statewide SLF scouting program that we run. So all of our vineyards within the program, and again, there are 400 plus vineyards in Ohio. We sample about 30 to 35 of them. But all of those blue dots are our distribution of our vineyards. And you can see they're pretty heavily located where a lot of our grape acreage is, but we try to cover as much of the state as possible. And again, this program has been really great because it's been running from 2022 and we're just picking it up again this year. So we have a lot of really important historical data on these sites, including when they were first testing positive, if they tested positive, and whether or not they continuously test positive for SLF. So at each of these sites, we compared our 3 different monitoring methods. Once again, it was those circle traps, that in-person scouting, and those eDNA collections. I'm going to go over the different results that we have. Again, this is a little bit of a busy graph. So on that x-axis, we have all of the years of our data collection, 2022 to 2025. And then we have the percentage of positive samples based on the trap type. And we're going to go through and we're going to talk about some of the different trends that we see. The first thing that we really want to point out is that the trapping and scouting have really low detection rates. So that's that purple and the yellow that you see there. I do want to point out that for the 2022, we never saw any SLF in any of our vineyards. And then in 2023 and beyond, we were starting to get things in the circle traps, but really those are low percentages. In the 2022, We do have 25% of our vineyards positive, but that was our flagship year of the program. It's our pilot program. So really 25% of 8 vineyards, 2 of our vineyards we were ever seeing SLF. That actually holds true for the rest of 23 and 24. It looks like the scouting dropped, but again, our vineyards increased. So we're still sitting at only about 2 to 3 of our vineyards every year where we are actually visually seeing SLF. What we do see, however, is that eDNA, the same thing that we saw in that urban study in Cleveland, really outperforms any of those other scouting methods. We do want to point out that what we see from 2023 to 2024 is a really big jump in the number of SLF vineyards that we're having positive for that eDNA. And while this is kind of alarming for our growers and they were pretty worried about it, what this meant is those eDNAs that we had positive in 2024, the scouting data is low, but learning that those vineyards were positive in 2024 allows us to reach out to the growers before 2025, before the field season, and help prepare them for what that infestation may look like. Whether or not that was 1 or 2, eDNA blips that showed up, or whether this is currently an active infestation, this program was really helpful for getting our growers ready to scout for SLF, learning where it's at in their vineyard. And for 2025, we're still analyzing a lot of those eDNA results just because, again, we went from 22 vineyards to 30 plus that year. So there's a lot more sampling that had occurred. But it goes to show that our growers found this program really valuable. Every year they continue to participate, and we're so thankful for them. So the question is, for these positive eDNA results, where in the vineyard are we getting these eDNA hits or these eDNA detections? And there's a couple places that we are looking for them. First, we're looking in the vineyard. Of course, we care about whether or not you have SLF on those vines themselves. This is going to be really important for any of our scouting because the vines is what we are worried about with our growers. It's really helpful. We know that SLF is an edge pest, so we can make sure that our eDNA scouting is along the perimeter of the vineyards. We're not going into them to scout for them. It makes our testing a little bit more efficient. We are also taking samples, eDNA samples from the landscape. So we know, again, that tree of heaven is such a huge contributing factor to the establishment of SLF. We're making sure that we're taking time to scout those trees, that we're running our DNA rollers up those trees, making sure that if they are present, we are searching for them as well. And the third place that we wanted to spend a lot of time looking was those transportation corridors. So we know that we've seen SLF become this really human-mediated pest movement, but with vineyards, it's a little bit different. A lot of our vineyards, we have a couple that are urban, but in general, these aren't sitting in big cities. We don't really have any vineyards that are in downtown Cleveland or Columbus or Cincinnati. And so what we focus on when we're talking about transportation corridors in vineyards is really parking lots. So where are people coming from? Where do they come in? So not just like the back of the vineyard, that landscape area where you push up against the woods, but also making sure we're paying attention to the front of the vineyard where people sit and they come in, they park their cars for extended periods of times because they're sampling the wines. We want to make sure all of that is covered. So what we found is that in our samples, about 63% of those eDNA samples that were positive were coming from the landscape. So 63% of them are in the landscape only, which means that they're not in the vines in Ohio. This is going to be really important. Again, we had already talked about, Ashley touched on the fact that we see SLF moving from the tree line into the vineyards. And this is really kind of confirming and supporting a lot of those theories that they're coming from the landscape. And then you have a little bit of time before they're in your vines. But what we then also see is about a third of our vines that we tested were positive for eDNA. What's really interesting, however, is that those vines that were positive They were not at the back of the vineyard. They weren't along the sides. They're at the front of the vineyard where they're right at the parking lot. So some of our vineyards in Ohio, they're so beautiful. You go in and you park and they're this kind of immersive experience. You're right in the vineyard. But those are the vines that we are seeing that SLF eDNA detections really light up. And again, this sounds a little bit nervous and a little nerve-wracking for our growers, but it means that with this information, they can choose where they want to apply their insecticides, where they want to scout. They can remind their visitors to be mindful of movement and checking their cars before they come to the vineyard or as they're leaving. So again, while this information was not fun to give to anybody, it's been really helpful in being able to target our kind of management and monitoring systems. And so, of course, we can't see eDNA. And the question is, well, how do you see what isn't there yet? And again, that's the paradox of this invasive species, is our growers, they need to be able to know how to scout for SLF even before that they have it. And so what we've done is a lot of this 3D printing technologies. This is really great because it can get growers some hands-on experience with some models without moving any SLF to them, which we can't do. You can't take SLF to move it to show them. You can't take the egg masses to show them. And so with our 3D printing, what we've really done is target a lot of those egg mass stages. So if you've seen the egg masses, anytime you Google them, the descriptions that come up are, it looks like mud, it looks like putty, it looks like cement or lichen. They look like other species eggs, so that's spotted lanternfly in the box, in the yellow box, and then spongy moth underneath it. And so we're stuck in this position where you're telling a grower, somebody who has a really important, really vested interest in being able to detect this pest, that it doesn't look like anything. The eggs don't look like anything, but they simultaneously look like everything. And so what we've been able to do is create these 3D prints for training our growers or master gardeners or any stakeholders really to help search for SLF. So these are some of the examples. These are kind of our first iterations, but they go on this keychain there. And then as a grower is moving through their field, if they think they have SLF, they can reference their keychain, as well as those QR codes, which will take them directly to our Ohio Department of Agriculture website with tips on management and monitoring. Beyond, though, just giving out these key tags, we set up what we call the SLF scavenger hunt. So we have the egg masses, which don't just come on keychains, we have them printed as their own individual pieces. And we set up this scavenger hunt where we stick them right on the different tree trunks or on some infrastructure. Occasionally we've stuck them on the underside of like park tables and park benches, anywhere that we know SLF is going to lay its eggs, which is, again, everywhere. So we spend some time teaching our growers and our master gardeners, anyone participating, how to look for SLF, what those egg masses look like, where they should look, again, anywhere above the head and below the knee, really focusing on what we know is difficult to find. After this kind of in-class presentation, we send them out there to go ahead and look for those egg masses themselves. We have a whole Qualtrics survey set up. So what they're doing is, as they're searching for SLF, they can take pictures of the egg masses they're finding, which lets us have some really great information on whether or not they're finding the egg masses, how many they found, whether or not they were missing any. And again, we know from this study that the egg mass is most likely to be missed for anything below the knee or above eye level, which then allows us to kind of rework our program into something that is more helpful. At the end of the program, we find that our participants really seem to like it. We found that, importantly, they learned new information in the program. They felt good when they left. But perhaps most important to us was that they wanted to use the information that they learned. So not only did they like it, they are planning to use it, which for extension, I feel like is always the goal. We spend the time making the products, we develop the slides, and we want to make sure that everybody who attends is actually getting something useful out of our programming. After these training exercises, we found that 100% of our participants can identify SLF adults. This is not too surprising, but we're really happy that this is what we're seeing. Again, those adults are pretty distinct. They have that nice pink wing pattern. They have those really distinct behaviors in the field. But really important, we see that about 95% accuracy with those egg mass identifications. So that's really good. That's what we want to see. The egg masses, again, are some of those hardest life stages to identify because they look like everything and they look like nothing. And we're finding that with about 86% accuracy, we can identify, or our growers or participants can identify signs of SLF. So a lot of scouting is based on this idea. A lot of that in-person scouting is that you're finding SLF because you're in the field at the same time that it is, which doesn't always occur. It very rarely occurs. So being able to do something like identifying those signs, even if you're not seeing the insect, is really important for accurate scouting reports. And so to kind of summarize everything that we have learned over the last couple of years working in this program with our growers is that we know SLF is spreading in Ohio. We see it with increases in that visual scouting, but we really see it with those big jumps in that eDNA detections. We also know that the eDNA performs really well. It works really good. We have that kind of year-long gap before we see an uptick in actual visual sightings based on the eDNA. But this requires a lot of time and research and money, and you do need to have that really special skill set to go out and collect the samples and then run them back in the lab. We also know, based on the participation from our growers, that they're really interested and motivated in continuing to be part of this program. Again, our scouting network has grown from 8 to 30 plus in the last 5 years, and we're constantly getting requests for more and more. So our growers care a lot about this project. They care a lot about finding out where SLF is coming from and what to do once it's there. We know that the stakeholder education is going to be critical. So again, where do you scout based on some of those results? How do you identify SLF? Can you identify it by the signs? And then we know that that 3D printing improves that stakeholder scouting ability. Again, we saw that big jump in accuracy. We know that this is a helpful program, and we're excited to keep working on it as well. I do want to say that a lot of our eDNA scouting, specifically where in the vineyard we're getting those positives, we're starting our 3rd year of data collection on that project, so these results are preliminary. We're continuing to refine our program and work through some of those results, but these are kind of that preliminary early process results that we are seeing. And so I do want to say thank you to all of the techs and the extension agents who have worked on our project, all of our different funding sources, both for the eDNA and the 3D printing, as well as all of our growers who continuously let us in on their vineyards to tour and to scout. And then if you have any questions, we'd be happy to take them now. And we also have your credits to scan. Thank you.
Thank you, Ashley. Both of you. Okay, so we've got, I don't know if you want to both handle questions together. We both got quite a lot of them here. So let's see. So I'll let Natalie and Ashley get oriented there around the mic. Our first question is, the request was, could I get a copy of the intro spotted lanternfly on the tree video? I think where the person was putting their hands on the tree. That's pretty impressive. And I don't know if you're willing to share that if people email you.
Yeah, just email me and I can share the link. I think it's actually available on YouTube.
Oh, okay. Thank you. I had not seen that one before. It was impressive. Our next question was, could you explain how the eDNA is collected? Sorry if I missed this.
The eDNA collection is a little bit difficult. So collecting it from the field is simple. SLF, it emits the honeydew. So as it's feeding, it feeds so much that it is literally pooping at the same time. And as it passes through the gut, it sheds that DNA material. And so what I do is I go to the vineyards and I use a paint roller, like a literal long stick paint roller, the type that you would use to paint the inside of your house. And using that and the roller covers, what I do is you really swab the trees and the vines, looking to pick up a lot of that eDNA. And then what happens after that is it's taken back to the lab, and that part's pretty technical. I wipe it off the rollers, and then we run it through some qPCR machines, and then we know whether or not we detected eDNA. One of the limitations of that is we don't know how many SLF are in the field. We just know that at some point there was DNA in the field.
Mm-hmm.
Thank you. Another question, how do the different detection methods for SLF differ in terms of cost? How might that impact widespread implementation?
The eDNA is very expensive to run. The scouting, if you're doing your own in-person scouting, that's your time and your energy. The circle traps and the sticky bands, you can buy the materials yourself to set up, and they're not very much, maybe $10 a trap max for materials. The eDNA, however, is very expensive to run. It's about $35 to $40 a sample, and we're then taking 6 samples per field 3 times a year. So that price of the eDNA adds up very quickly. And so while it is very effective, it's very costly.
And I think it's a matter of pick your limitation. So if we're talking about vineyard managers that might already be walking their blocks, the easiest, I'm going to put this in quotations, the easiest is to continue to do that and look for spotted lanternfly, right? You don't necessarily need to set up a tree trap. You don't necessarily need to do eDNA. But that's from the perspective of someone who might be already walking their blocks looking for pests. If we're utilizing this type of perspective more in an urban setting, and we aren't maybe scouting a park on a weekly basis for spotted lanternfly, then a tree trap might be more accessible and easier for that person to kind of more passively sample. So I think it's very dependent on what type of system you're in and what types of what you already have, what resources you already have available to you, and kind of weighing the pros and cons. Because I don't think any of these approaches is the clear winner, is what I would say.
I'm assuming the eDNA is not commercially available. I mean, even if it were commercially available, do you think that would be helpful to growers or?
So that is something we are weighing. I know that there have been groups out of Virginia Tech, Tracy Lesky and Scotty Yang. I don't know if anybody related to that group is on this webinar. If so, certainly speak up. But they've looked at how we can maybe have a simpler way of sampling spotted lanternfly eDNA through actually sampling the ant population. And then there's been discussion of looking at a lamp assay. So that would be something in field, a lot easier, hopefully. And I say easier because it's probably gonna be very expensive. I shouldn't say that, but I think it will be kind of pricey. But the idea is then to be able to conduct that eDNA sampling without having to go to a lab. You can just go ahead and do that in your field.
Nice. Our next question was, can we install a sprayer of some type for entry in the parking lots that might be effective at destroying the spotted lanternfly as people visit the wineries?
I think it's important to remember that for the growers, a lot of visiting the winery is about creating this really unique experience. You're in the vines, you're out in nature. I think you kind of have to balance that consumer perception of spraying. So a lot of our growers, when it's the peak wine season, they're spraying at midnight so people aren't, exposed to those chemicals. People aren't seeing their foods being treated with insecticides. So I think that would be nice. We do have some growers that as they've become positive around their parking lots, they have asked for what we call CM Stomp-'em posters, which they kind of just staple onto their little telephone poles or on the doors. But I think that the reality of working in the vineyard and the wineries with tasting rooms in particular is that you have this really interesting intersection of this is one of the only crops where people are right there in the growing system. They want to be hands-on. So while I think our growers would really love to do that, it's really hard to balance that perception of insecticides with actual use of them.
I mean, so good follow-up question. What can a grower do after finding spotted lanternfly? Are they spraying? Are organic growers have any options?
Yeah, so I think we're at a really interesting space right now for that exact question. And this is something Natalie and I talk about on probably a weekly basis almost, because we are having vineyards show up as being eDNA positive. Maybe they're even seeing an adult in the vineyard. And now the question is, when do I need the care? And so I think that there's kind of two different ways that we can answer this question. So the first thing that I always say is it's very dependent on the grower and the stakeholder in terms of how they want to, what types of quality they want to maintain, or what type of perception, as Natalie was talking about, that they want to maintain. And maybe it's this pristine vineyard that never has SLF. I would say that that's not probably the best idea, because that means we're most likely going to have to intervene a lot and make insecticide applications that might actually cause more problems down the road in terms of secondary pests. But that is something that we discuss, is like, that is, we are dealing with this infestation. It is not our right to say what is best. I will tell you that there are thresholds that are being developed right now in the Northeast, and it looks like working with a threshold of about 5 SLF per vine is the threshold that's being proposed and tested. So that's kind of what we have been discussing with growers as well, is maybe we can wait a little bit to intervene until we see more of that population. But if you are really, hey, I want to spray, I don't like that I have any SLF in the vineyard, we would recommend a perimeter spray if possible, because we know that that's where the majority of spotted lanternfly is hanging out most likely. We also know that there was a publication, I think it was 2-3 years ago, that shows that this is an effective way to managing spotted lanternfly infestations, even when compared to a full block spray. Did I answer all of the facets of that?
I think organic.
Okay.
I think we already did.
So Natalie, do you want to talk a little bit about actually what you're doing this summer in terms of looking at some of the organic management options we might have available? I will tell you just real quickly that typically when we're looking at best performing products, it tends to be neonicotinoids and pyrethroids, occasionally carbamates like 7 or carbaryl perform best. But that does not mean that we don't have potentially some organic options that might be effective.
This is something that we are just starting this year, is looking at, now that we have, SLF in Ohio, is really looking at what products that we have available, what's their efficacy, how long they're going to last. Right now, we don't, unfortunately, have any results from that because it is peak SLF adult season. For organics, again, it is, I think, a little bit more difficult because a lot of those studies have focused on some of those really conventional products. But from what I've been told, speaking with growers, they use a lot of like the mineral oils to coat the trunks of their trees, but a lot of that's going to be done in winter because you're looking to smother a lot of those egg masses. But I've also been told from growers themselves that specifically we have some that are in right up against residential neighborhoods who have a lot of restrictions on their spraying, and they told us that they occasionally just fill their tanks with water and they just spray SLF straight off the leaves with no products, because if those wings are damaged, they can't really do much and the wings are a little bit touchy. But right now, I think unless you can think of any, we don't have a lot of organic products.
And it's mostly entomopathogens being the primary, is looking at some of these fungi that have come out more and more. Obviously, we have what Ann Hayek's group put out. I think it was like 6 years now, maybe even 7 years, looking at some of the different entomofungi that can be pathogenic or cause mortality in spotted lanternfly. And even more recently, that's something that's continuing to be proposed for actually causing spotted lanternfly mortality and having kind of more of an OMRI or organic perspective approval.
I'm going to the next question, I'm going to translate a little bit, but they're asking about when growers are doing scouting and things like, are you getting that information? Are you including that in your data when people other than you are doing scouting, like are using a platform or EdMaps, or how's that working?
So, yeah. So we do a lot of the scouting ourselves, which means that I'm out in the vineyards. But of course, the growers, I go out once a month because we have so many. I can't be out there every week. We would like to, but we can't be there every week looking at every vine. So we do work a lot with the growers to do the scouting. The growers themselves spend a lot more time in the field than I ever do. Of course, like that's what they're out there for. So it's a little bit of a mix. What's been really exciting for us or for me is that I do get calls from our growers on the weekends, after work hours, and sometimes they do see SLF before I do, and that's been really helpful. That is data that we are tracking because as the scouts that go out once a month or every 2 weeks, again, it's like searching for a needle in a haystack. I have to be out there the same time as the SLF, and the time that I'm out there, if it's only a couple times, a season, the growers are much more likely to find it. So we do have a lot of that information. We have the dates that the growers found them, the life stage. A lot of the times they do send really good pictures or they save the samples for us. So that way we can confirm what they are looking at and making sure that what they think is SLF, which with our training they've been they know exactly what they're looking for. So it is definitely a team effort to make sure that we're covering every vineyard as much as we can.
Okay, we've got a lot of questions. The next question is the DNA of the food the spotted lanternfly ate, or is it part of the lining of the gut?
Okay, so I think there might be some confusion. So when we are doing the eDNA sample, it's basically environmental DNA. So what we're doing is we're sampling the landscape with the hope that whatever we roll, we are going to pick up spotted lanternfly DNA. So spotted lanternfly in animals generally, when they defecate, will shed their DNA. So that's what and granted, that's a working hypothesis. I should also acknowledge that. It's also possible that there's other things that we're picking up that spotted lanternfly is shedding in the form of its DNA. We assume it's most likely the honeydew that's being produced or the poop because we see so much of it. So that's most likely what we're seeing in terms of where that DNA signature is coming from. Whenever I'm explaining this, I often say, and that's why we didn't have the time to kind of share that, but we kind of jokingly talk about it as being like a CSI episode for invasive pests, because what we're doing is we're kind of like scrubbing the environment for the culprit's DNA in the same way we would kind of process a crime scene. So we're doing the same thing, except for the criminal in this case is spotted lanternfly. So when we're thinking about what we're picking up, it's not so much that the DNA is in a specific spot, it's that it's defecating and shedding that DNA. Hopefully that makes sense.
Okay. We have a couple questions on kind of how quickly the eDNA breaks down. Is there a possibility that you're picking up like 2-year-old eDNA? Do you know?
So we know that eDNA breaks down within 10 days, faster depending on the UV or the rain. And so going every 2 weeks, though, making sure there's a minimum of 14 days between that sampling. And really, as we picked up more sites, going once a month means we're not sampling that same population. So any of those eDNA hits are new to that environment.
Okay. We had a question about damage to the vines or the trees. If you could just describe it really briefly, if not, like a good website people could check.
Yep. So spotted lanternfly damage is very difficult to see because it's a phloem feeder. It's a giant aphid, essentially, is what I like to say, even though it's not an aphid. But it has this big, straw-like mouth part that pops into the vineyard, the tree, or excuse me, the grape or the tree or whatever it might be, and then it's going to feed on that phloem. That's going to cause stress to the plant and potentially cause the negative impacts that I shared with the grape specific example. And that can cause, in some cases, leaf redness, depending on what type of plant we're talking about. It's also, I got distracted by the other question. It could also be looking at honeydew deposition, so that shiny poop that's great, that SLF is actually pooping out, that honeydew can actually be a medium for sooty molds, and that can be a photosynthetic block, so that can be something that actually causes leaf dieback. We can also see wilting in some really severe cases. So it's not something where, a lot of times I say it's not a locust. You're not going to see chewed up leaves, for example. You're going to see really plant stress as being the primary stress, or the primary symptom.
So the next question is Ohio suggesting systemic trap tree treatments. I don't know if that's a tree like tree of heaven treated with systemic insecticide. I guess I'm not aware.
So that's more of an Ohio Department of Ag question. I would not, that is something that is actively in the works in the Northeast, is looking at some of these trap tree treatments and maybe integrating that within a vineyard system. I will tell you that the data looks very good on that, but there are some kind of concerns in terms of what systemic insecticide we're using when we're applying it, what that might mean for the beneficial insect community.
Very good. Next question was, can you spray clay pesticides?
So I assume we're talking about surround. And I would say that does have some efficacy, but it's limited. So it's all dependent on, once again, if we're able to have a thick enough coverage. Whenever I'm talking about surround, it's basically the same conversation, making sure we have enough of that material on the plant, that we're not creating any type of problem with the actual plant growth, but also interrupting the ability for that animal to actually feed. I will tell you that in grapes, it'd be very hard, I think, to get enough surround. In these bioassay or kind of laboratory results, it does show some efficacy, but that's a lab, not out in the field.
Okay. I'm kind of jumping around because I know we just have a few minutes left, but I do want to drop to the last question. Was it just mentioned that 63% of the eDNA is coming from the surrounding landscape? Would there be any point of doing like landscape treatments rather than in the vineyard, or any thoughts on that?
I think it's going to depend on the individual growers, and it's also going to depend on what they are legally able to do. Some of these vineyards, they back right up to, you're good, checking the time. They back right up to residential areas. Some of them back right up into state parks or protected woodlands. And so the management of spraying within that landscape, specifically any of those trees, removing tree of heaven, that's something that I think is a really gray area for a lot of our growers. And this has kind of been a discussion that we've been having with them, what they are allowed to do, what is not technically their property anymore. And so we would like to look at that landscape management. But again, the host range of SLF is so wide, it's going to be really difficult to remove a lot of those kind of host trees. And I think someone did ask to go back to the host slide. So I think I can pull that up.
Yeah, we did go through it quickly. That is a fair point, by the way. There are a different variety of different plant species that spotted lanternfly can utilize. This is not anywhere close to the full list of what we would expect. So when you look online, well over 100 different species that they can take advantage of. But the other thing I wanted to acknowledge that Natalie touched on, there are research efforts in the Northeast and Virginia in particular to look at more of an area-wide management approach and specifically targeting tree of heaven reduction with the hopes that it will create corresponding drops in spotted lanternfly. So that is an ongoing effort, is to understand how we can maybe interface more with the fact that spotted lanternfly has a primary host that it likes to use, which is tree of heaven.
Okay, very good. I'll just do the last question really quick. It's just asking about stages 2 and 3 causing damage, or is it just stage 4?
Okay, so all are capable of damage. They all feed, I would say, obviously not necessarily egg masses because they're just sitting there waiting to emerge, but the first instar all the way up to the adult is capable of feeding on plants, so causing damage. I don't think there's been outright data that have been released regarding grape production and any negative impacts of just nymphal feeding on grapes versus adults. And that's kind of the issue with that, being able to really say, okay, well, this damage was only caused by nymphs, this damage was only caused by adults. A lot of times it's you're getting all the life stages in that population. But yeah, they all have the same mouth parts. They're all capable of feeding on plants, so they're all capable of causing damage. Adults are the primary problem. Those are the biggest, and they remove a lot of sap.
Okay, well, thank you both so much, Natalie and Ashley. You answered a lot of questions. There's still a few more, but we want to respect everyone's time with the 1 o'clock deadline. So again, thank you. Great information. We will send out the video just to let everyone know our next Pest in Progress webinar will be October 21st at 12 p.m. Central, where Dr. Mandy Bish from the University of Missouri will be discussing current trends and management of plant parasitic nematodes in Missouri soybeans. So thank you again both so much. Spotted lanternfly are always a very interesting topic. So everyone have a great rest of your day.
Thank you so
Thank you.
much.